Chicago, but I'll be speaking to you today about some undergraduate research I did at UC Berkeley with Elizabeth Barrere. But before I get into the specifics about the study looking at clonal and bisexual whiptail lizards from North America and Mexico, first I want to acknowledge and thank some people for support, training, funding, museum collections, access, and specifically highlight Sarah Warning and the Calendar's STARS research program for getting me into science and doing this project. So thank you. This is showing the modified phylogeny from Pyron et al. 2013, which is a larger Swami phylogeny where the Teiidae is, and more specifically where the genus Aspidicillus falls within Teiidae. Different trees have found different relationships within the Aspidicillus genus, but later on we'll be discussing a tree that I'll be using, which we created with different genetic markers that's helping parse out some of the interrelatedness between some of the hybrids. But Aspidicillus is a useful genus for looking at comparing clonal and bisexual reproducing species, because approximately 1/3 of all the species within this genera are unisexual. So they're all female and they reproduce via obligate parthenogenesis. Previous work looking at morphological variation within this genus has been mostly focused on meristic scale variation, which is useful for species identification, but there can be a variation within species regarding these traits. The classic example of meristic scale variation that most people focus on is number of scales or scale counts. And here I just wanted to highlight two particular studies that directly compared unisexual and bisexual species within this characteristic. Taylor et al. 2012 focused on samples that were from same or geographically adjacent localities and found that A. tessellata, the unisexual species, was not always predictable based off a reproductive mode on the amount of variability relative to bisexual species. So sometimes it had greater variability, sometimes it had equivalent, sometimes it had less depending on which species we were looking at. And Cole, Taylor, Townsend, 2016 used clonal laboratory lineages of a unisexual species to compare that to bisexual field samples, and they found that they were as variable in all 10 univariate characters that they looked at. So this is very interesting and kind of goes against what you would expect with lower genetic variability maybe being indicative of lower phenotypic variability, and allows us to build upon this and look not only at univariate and multivariate scale variation, but go one step further and look at multivariate shape data, which is what I'll be showing today. So the key questions that I'm going to be addressing first is, does my clonal natural population exhibit more or less morphological variation within multivariate cranial shape data? And our goal for this study was that it would exhibit less. Then moving forward, do epigenetic factors such as year collection, field assessed age, sex, species, or combinations of these significantly explain the morphological variation between or among these species? Then what role or influence might size have on shape within our population? So can we pick up an allometric signal? And then can we pick up further parental taxon or phylogenetic signals within our shape space that might explain more of the variation in epigenetic factors? So getting a little bit more into detail about the particular species that were used for this morphological analysis, the specimen of interest for our parthenogenetic or clonal species was Aspidicillus felix, which is found in New Mexico. So our field site is right here in Ghosts Ranch, which is just northwest of Santa Fe. We collected species from a 50-meter radius off the red sandstone there to control for habitat heterogeneity, and that's just a photo of what it looks like. And then we compared it mainly to a bisexual species, Aspidicillus sexleniata, which was field collected from a similarly environmental homogeneous locality, the Walt Disney World Resort, which is what it's called. And then to kind of compare these broadly to how shape relates within this taxa, or within this genus. We compared it to not only the parental taxa, but also more species within the Mexico complex. And I particularly want to note Aspidicillus inernata, which we'll be looking at further, which represents a twofold paternal contribution in the hybridization event, which caused Aspidicillus. So the first part of the methods is first to look at how we measure shape variation. I did this using geometric morphometrics, which landmarks points across specimens to statistically analyze shape change among and within groups. So here you see both dorsal and lateral view were used, and I superimposed the Procrustes superimposition on top of the skulls to show you where the landmarking were. We used curves with semi-landmarks, accounting for bending energy. And yeah, we used 28 specimens and 99 amongst all of the other ones, and all the analyses were done in Jupyter. The second component of the study was constructing a more resolved phylogeny within these groups. Aspidicillus vilax has been thus far phylogenetically unresolved. So I included 24 Aspidicillus species and 7 outgroup taxa that were from previous studies for consistency. And I used 2 mitochondrial sequences and 2 ribosomal RNA genes and to get the most coverage and spread. And we tested for phylogenetic signal using the K multi-statistic from atoms in the GMOF package. So looking at the first question within our results, we first have to look at, does the clonal species have more, less, or comparable levels of morphological variation? You can see here that there's 2 PCA plots, one for lateral view, one for dorsal view, and they tell very different stories. We have here the morphological disparity measure, and then the p-value from pairwise comparisons, testing if these morphological disparities are significant. You can see that in lateral view, Vilax is more variable, in dorsal view, it looks like it might be slightly less variable, but the p-values are not significant, so I would say that these have comparable levels of variation, depending on which view of the story you're looking at. Looking at factor significance to explain our morphological disparity, we see that when we look at all the species included, including the Mexico complex, every single one is highly significant. But I just want to highlight 2 particular cases that show a different story. When you look at the dorsal view for Vilax insects lineata, including males, females, adults, and juveniles, you see that it's significant. But then when you look at each taxa individually, we see that it's actually not significant. So that's taxonomic conflation of factors. And when you look at within Vilax, particularly date collected, we can start to pick up population structure and differences in views where dorsal is significant and lateral is not. Testing for size influences on shape, we can do this by plotting size or centroid size from our data set regressed on shape. And to look at the allometry, we run an ANOVA comparing our shape and size expected relationship to ones under friendly motion. And we see for dorsal, just adults and only females from both Vilax and sex lineata, even accounting for species or taxon differences, it's not significant. But when you start including more and more taxa, the significance, even accounting for species, increases. We see a slightly different pattern when we look at just the lateral, and we also see overlap starting to happen within Vilax and sex lineata here, and then this would be the Mexico complex, where before there was separation. And in addition, even accounting for taxonomic groups within the analyses, there's a significant relationship between size and shape that's different from dorsal. This is the principal component analysis of the dorsal view of all of our species, showing where they're plotting out on the morphospace. And I just want to highlight a few of the different groups. So green is Aspidicillus vilax, blue I just highlighted this larger band to show the rest of the Mexico complex to give you a sense of the related spread between all of these different taxa. And what we notice here is there's some overlap between the daughter and the parent species that might be indicative of some morphological similarity within our measures. And this got us interested in testing for phylogenetic signal, but I do want to point out that within lateral there was no overlap of these species. So testing for phylogenetic signal, here we have a collapsed phylogeny from our created one, which has Ospidicillus vilax next to Costata costatus, not next to Inornata, and we'll talk about that a little bit more later. But here we have phylomorphous spaces, and you can see that closely related taxa are generally widespread across this phylomorphous space. And the phylomorphous spaces were constructed with the name shapes for each of the species, and the white nodes are the ancestor state reconstructions for each of the nodes. And in order to measure this a little bit more concretely and quantitatively, we used the case statistic where closer to 1 means more phylogenetic signal, and p-values that are smaller or more significant are compared to 1,000 iterations expected in a Brownian, and we find that we get a low k and a high p-value, which means that there's no significant signal for phylogenetic signal within our local space. And then this again is our phylogeny that we got from the reconstruction, and I just want to highlight a few things and drive home some conclusions. First off, cranial shape variation was not predictable based off of reproductive good within our species. Testing for influence of factors across and within groups provide more of an insight on the relative importance of each, and highlighting parental taxon, morphological overlap is not necessarily indicative of phylogenetic signal, but I'm going to have a caveat here because we had our Ospidicillus vilax fall out with Bertii, and I think that's because we included mitochondrial genes, and therefore it has a more maternal signal. So perhaps using other genes, you might get different relatedness, and you might have a stronger phylogenetic signal in that case. Or including Bertii, it also might have a lot of overlap in that, we can't say yet. And future work will involve histology to get exact ages and population structures between the years collected, to see if there's any change in that and accounting for the date collected significance, as well as a genetic component to look at different influences of locality between our locations.